Okay. So the purpose of my talk today is actually to present you a large-scale candidate gene approach I've been developing to tackle the evolution of associative mating, and in particular some work I did on the P aphid. So obviously we know that associative mating can result from the evolution of different types of traits. For example, mating preference leading to sexual isolation, ecological specialization inducing the evolution of host habitat preference and therefore habitat isolation. And for example, in plants, the evolution of flowering time leading to temporal isolation among plant species. So for the past few years, I've been focusing my attention on the mechanisms and factors that are favoring the evolution of associative mating, specifically in the face of gene flow. And this in different study systems that are actually representing different modalities of premating isolation and of gene flow. And for all of these cases, actually, one factor which is influential for speciation is actually the genetic basis of bias to gene flow, of course. And so far, I think two main strategies have been developed to address this question. The first strategy is to run some QTL or association studies that will be using phenotype and genotype information data in order to identify loci explaining variation in traits. And the second strategy is to run genome, or more recently transcriptome scans for selection using only genotypic information, and in order to between diverging population, of course, in order to identify outlier loci. So today I'm going to present a candidate gene approach which actually represents an alternative route to the genetics of speciation, and which hopefully can complement the QTL and population genomics approaches. So, as chemical senses are actually frequently involved in at least some aspects of associative mating, and this is very clear in various insect and vertebrate species, this implies that actually we can, the chemosensory genes can be good candidates for a role in associative mating in many systems. Chemosensory genes being genes underlying chemoreception and pheromone production. So having these candidate genes in mind allow formulating some clear and testable predictions. If some of these chemosensory genes are involved in associative mating, they should show some patterns of divergence among different populations and possibly divergence under selection. So among chemosensory genes, we can find genes encoding odorant binding proteins that are involved in the stabilization and the transport of odorants, and that sometimes can play a role as pheromones themselves. A second class of genes are genes encoding for chemoreceptors that are located in the dendritic membrane of chemosensory neurons, and that interact directly with chemical signals and translate this interaction into an electrical signal, which is going to be processed by the central nervous system. So as new genomes are being sequenced, the structural similarity of these proteins across various phyla and different invertebrate and vertebrate species is being revealed, and also is being revealed their genomic organization in quite large multigene families in most genomes. And this latter point implying for us to develop some large-scale candidate gene analysis. So as an example of application of this type of approach, I'm going to talk about some work I did on the PA feed, which is a model system for studying ecological speciation. So pioneer work done by Sarah Vaya and collaborators on North American host races, which are specialized on alfalfa and red clover and that occur in sympatry, has shown that host plant specialization is actually a key component of reduced gene flow in this system, as it induces selection against immigrants, selection against hybrids, and also associative mating as a result of host plant preferences and local mating on these plants. Moreover, we know that genetic studies has evidence or provided some evidence of pleiotropy or close linkage among loci underlying host performance and host preference. This suggesting that maybe host preference in this system is evolving under kind of direct or pseudo-direct selection. These patterns have been recently confirmed in a series of European host races and species, and nicely showing actually a continuum of differentiation among these different biotypes. And we know in the PA feed that host plant acceptance is actually a question of smell and taste. So here the aphid is using this antennae and it's probing the plant tissue, and this is the key step for host acceptance in aphids. So because of this information, that's the reason why we focused on these chemosensory genes as candidates for associative mating in PA aphids. And so we, for actually more than a year, we worked on the molecular characterization of these genes in the PA aphid genome and identified a number of odorant and gustatory receptors, odorant binding proteins, and their related chemosensory proteins. So with this sequence information in hand, we could therefore address the role of these genes in host plant specialization by testing for some divergence of these genes under selection among different host races. This work has been done by, in collaboration with Julia Ferrari, Jing Jiangzhu, Bjorn Kembach, and Roger Butlin. So tackling sequence variation at numerous loci, and this at the population level, is obviously a real technical challenge. And we decided to use the recently available sequence capture technology in order to scale up this candidate gene analysis. So the sequence capture experiment consisted in extracting DNA from 8 individuals of 3 different European host races that are closely related but highly specialized on different host plants, namely lotus, alfalfa, and red clover. Then designing the capture array with sequence information for more than 200 candidates, but also control genes randomly chosen in the genome. Then capturing and amplifying these targeted sequences, and finally sequencing these captured fragments using 454 technology. The sequence capture experiment worked pretty well, maybe surprisingly. Here I show the capture specificity, which is actually the proportion of reads that mapped to targets, and it's around 50%. It could be relatively small, but actually that's what is to expect with this nibble gen technology. More importantly, it's comparable among the different samples we studied. The capture uniformity is also very good because we managed to actually capture 95% of the targeted genes and 90% of the targeted exons. And actually the one missing were actually the ones that were poorly annotated in the first version of the genome assembly. So it makes sense. So we developed then a bioinformatic pipeline in order to process this capture data from raw reads to some population genetics estimates, and these going through different steps, assembly and quality control steps, and SNP calling, selection, and analysis steps. One issue we had to deal with was the estimation of allele frequencies and population genetics estimates from next generation sequencing data, as this technology is actually introducing some biases due to a high sequencing error rate and also an uneven chromosome and or individual samplings when we use pooled data, which what we did. So in order to get rid of this problem, we actually applied some bias corrections proposed by Fuchik and Schlotterer, which consist in setting up a minimum allele frequency for the SNPs under analysis, correcting pi estimates using read coverage, and averaging over multiple sites. So I just got some data on polymorphism and divergence at these candidates loci not later than last week. So this is, let's say, work in progress. But I thought it would be nice to show you some results today and maybe discuss this result at this conference. So we used nucleotide diversity, pi, within and between samples as a proxy for differentiation. And here are plotted FST values for all of our genes each of a given length. This in a comparison between lotus and alfalfa host traces, the color code representing the different gene families we've been studying. So we can see that there are some quite high values here, FST values for some of the genes, which can be compared to the average FST at neutral loci. And if we look up there, we can see some loci that are highly differentiated between these two populations, and that correspond to an odorant receptor and two gustatory receptors. We can just repeat the same type of analysis for the other pairwise population comparison, and again visualize some highly differentiated loci among these populations. However, the FST is actually probably not a very good statistic to compare divergence between population, as it confounds the variation within and between populations. So I decided to plot instead diversity between samples as a proxy of differentiation, and this is plotted against diversity within sample here. So again, we can see up there some loci with high differentiation between lotus and alfalfa, and using some permutation tests, we can actually show that two of these loci that are actually odorant receptors are significant outliers. We do the same for the other population comparison, and we identify 3 odorant receptors that are highly differentiated between lotus and red clover, and no significant outlier between alfalfa and red clover. In a second step, we used the ratio of non-synonymous to synonymous diversity within and between samples as a proxy for divergence selection. And using the same procedure, we can identify some loci that show some signature of divergence under selection between these different host races. So this seems to be a quite successful route, this large-scale candidate gene approach, to narrow down loci of interest. The combination of summary statistics allows the identification of a handful of loci with known function that show some strong differentiation and patterns of adaptive evolution. Combination of pairwise population comparison actually indicates that most outlier loci are common to all comparison, which can be used as a cross-validation, but can also indicate kind of universal loci involved in host acceptance. And finally, the combination of different types of candidate loci seems to indicate that odorant receptors, and to some lesser extent gustatory receptors, are of primary importance in host acceptance in aphids as compared to other type of chemosensory genes. So of course we want to follow up these first results with further sequencing, maybe improved population genetic estimates and tests for collocation of these loci with already known QTL. And a current project, NERC-funded project coordinated by Roger Butlin, is actually aiming at scaling up this study by increasing the number of host trace comparisons and the number of candidate genes. It also aims at tackling variation in more dimensions than just the sequence dimension by adding some expression and copy number variation information. And in the longer run, we may want to develop some functional studies to understand how these loci can imply behavioral divergence, and also maybe test for the role of non-genetic mechanisms, such as ecological imprinting in the evolution of host plant preference, this in relation with expression studies. So before wrapping up, I just want to briefly mention that I'm developing the same type of approach in another fascinating system for speciation research, the house mouse, for which we observed olfactory-based isolative mate preference in the hybrid zone, which could be related to evolution at some chemosensory genes, major urinary proteins and vomeronasal receptors. So the idea here is again to test for some differentiation at these loci between different subspecies, and also to use these candidate genomic regions as a way to seek for signature of reinforcement among mice populations. So this is a project just starting, and there is a poster presenting the project a bit more generally. So I'd like just to finish off to thank all of the collaborators on this project. Don't interpret the size of the aphids here. And thanks you for your attention.
Questions? So I have a question that relates to studies of this sort.
Sorry, I can't hear you.
I have a philosophical question, I suppose, that relates to studies of this sort. And I hear Louis Burnish shaking on your ear right now telling me this. And that's, how do we know, and how do we decide when the genes we're looking for are speciation genes or adaptation genes? Are they the same thing? Are they different? And if they are different, how do we figure out which is which?
Yeah. Yeah, well, that's obviously a question we all have in mind when we do some genomics of speciation. So in this system, we start from the phenotype, and we know that actually this host plant specialization is involved in the reduction of gene flow. So that's the starting point. And then we followed some candidate genes, which actually can be involved in adaptation and in reproductive isolation, but even more than that, can be involved in different components of reproductive isolation. So at the moment, we are assuming that they are involved in host plant preferences. I also have in mind that they could be involved in sexual isolation among aphids, right? So these different, to tease apart these different things is probably a question of modifying these genes and testing what is the impact of the modification of the genes on a specific behavior. And this specific behavior could be either a sexual behavior or a preference for a plant or something else. If it's not related to any trait that is involved in premating isolation, then it's not related, associated to speciation. That's the...
in ecological speciation, are there any genes for adaptive versions that are not also genes for reproductive isolation?
Yeah, there must be.
Like what?
In this system, you mean, or in general?
Well, in any system, really. I mean, if adaptive divergence causes selection against migrants and selection against hybrids, then every gene, I'm just philosophically asking.
Yeah.
Because then every gene influences performance on the opposite poles. Every gene for adaptation.
Yeah.
If that's the case, then all adaptation genes are also speciation genes.
Yeah. Well, yeah.
Maybe this is best discussed a little bit further in coffee break, unless you want to try at the Hi, this is actually a really good question. You focus on, you talk on the genetic, on the variation of genetic. So maybe asking about the impact study out there. So I'm curious. So you showed one of those plots where I think those are alpha and divergence between, so high between species and high species. And I noticed that actually the maximum divergence actually not there on the same scale, so one choose in your show. And I'm curious as to how much genetic variation and potentially phenotypic variation are there for one of those. So, are they within the species really having the capacity to actually try out different software, different kinds to start with?
Yeah, so that's a good point. I have to say I haven't really fully analyzed the data, but you are right that here, it's here, yes. Pi within is quite high. Is that what you mean? Yeah. So, So, of course, the importance is the relation between that and that, right? But it's also the absolute value of this. And obviously, there is this pi within which is quite high, which can be due to many factors. So, it's difficult at the moment for me to interpret this. So, Charles Ross actually and collaborators have shown that pi within is also supposed to increase when pi between is increasing. So that could be a side effect of differentiation among populations. But that could also be due to many other factors specific to each of these populations. So I don't know yet. My important point is that these are still quite high pi between values, and that's my interest here. But you're right, yes.
Okay,